Studies done inside the laboratory use pure isolate, and so everything we work on and with, like the surfaces, the glassware, and media, should be sterilized. To prevent other microorganisms from contaminating pure cultures, we use procedures called aseptic transfer techniques. These also provide protection for you and your environment from microorganisms. We have to wear appropriate personal protective equipment or PPE, namely sterile gloves, lab coat or lab gown, surgical mask, and goggles. Now, if your hair is long like mine, please do tie it. Do not wear these PPE outside the laboratory to prevent the spread of pathogenic microorganisms. Prepare your glassware and tools. They should be washed and dried, ready for sterilization. Cotton plugs are used to seal flasks or test tubes that contain media. These are used to filter out contaminants present in the air that enters the flask or test tube, and are specifically used for agar slants or isolates grown in solid media for storage.
We interrupt our program to bring you this important message.
How to make cotton plugs. Step 1. Prepare your materials. Step 2. Take your cotton and cut it to size. Step 3. Roll it as tightly as you can, and then add more cotton if necessary. Step 4. Wrap the rolled cotton with gauze, and the cotton plug must be sturdy enough to be easily inserted into the opening of the tube or flask. It should also make a popping sound when removed. And step 5. Tie the cotton plug with some cotton string to hold everything in place. And there you have it. You have your very own cotton plug. The sudden change in temperature and pressure inside and out of the sterilization vessel may cause damage of some glassware or materials, so make sure to wrap all necessary materials like loops, glass slides, glassware, etc. with paper or foil. This is also to prevent further contamination of these materials outside of your sterilization vessel. To wrap petri plates, you may use new or scrap paper. So you have to lay your paper flat on the surface in landscape orientation. Next, place your petri plate at the center. Then fold each shorter side of the paper until they overlap on top of the plate. Now fold each corner at an angle, and then finally, fold the flaps toward the bottom of the plate. We recommend that absorbent materials such as cotton plugs and cotton swabs be wrapped with foil to prevent them from getting wet during sterilization, as this process involves steam. Place materials in an autoclave or autoclavable plastic. Seal the plastic with a rubber band. And place it in an autoclave basket and then into the autoclave. Turn the autoclave on and let it do its sterilization magic. This process should take around 2 hours.
2 hours later.
While waiting for it to finish, turn on the UV light in the Biosafety cabinet. This should take around 30 to 45 minutes, and then you're ready. So, I would like to introduce you to the Biosafety Cabinet or Biological Safety Cabinet. This is designed to protect you and your environment from pathogens. Before beginning any activity in the Biosafety Cabinet, wipe the surface with disinfectant or rubbing alcohol. Prepare the labels for the tubes to be inoculated with the name of the bacterial culture and the date of the activity. If the isolate is stored in liquid media, gently mix the stock culture for uniform distribution of the cells in the broth. Then secure the stock culture tube and the tube to be inoculated in the palm of the less dominant hand, in my case, the left hand, and secure it with your thumb. The tubes will form a V shape in your hand, and this is to prevent direct exposure to airborne contaminants. Using your dominant hand, take the inoculating loop and flame sterilize it. In this case, we are using an incinerator. When using the alcohol lamp in flame sterilization, just flame the loop over it until it turns bright red. Once sterilized, never put the loop down on the surface. Instead, return it in the tube containing alcohol. Using the same hand holding the loop, remove the caps or plugs from the tubes and hold them between your fingers. The inner aspect of the caps or plugs should point away from the palm of your hand. Flame the lids of the tubes and take an inoculum from the stock culture using the inoculating loop. Insert the inoculating loop into the subculture tube. For a broth medium, shake the loop in the broth to dislodge the cells, and for an agar slant or solid medium, introduce it in a zigzag or fishtail manner. After inoculation, flame the lids of the tubes and replace the caps or plugs in the same order. Flame sterilize the loop to kill remaining microorganisms and place it back in the tube with alcohol. Then wipe the surface with disinfectant and turn on the UV light. For sterilization of the Biosafety cabinet. Finally, label your tubes and place the tubes in autoclave or autoclavable plastic and incubate it at 37 degrees Celsius for 24 hours. After recording necessary results from your subcultures, make sure to decontaminate them in the autoclave and throw waste in their appropriate containers. Proceed then to washing and drying the glassware and other materials used for the next activity. Make sure also to wash your hands every after laboratory use.